And then you see the serum. Serum meaning it is the blood plasma from which fibrinogen has been removed. Fibrinogen meaning a kind of protein that is dissolved in the blood. When at the time of blood clotting, the clotting material, that fiber like structure found in the blood after its clotting, that thread like substance is formed from the fibrinogen at the time of clotting. After removing that fibrinogen, the remaining liquid part of the plasma is known as the serum. Sometimes actually blood plasma from which fibrinogen has removed is known as the serum. It may contain numerous chemical substances including antibodies, antitoxins, etc. You know that usually at the time of snake bite, we use the serum developed from the horse. You might have heard about it for the snake bite, especially for the snake bite, for many diseases. In certain cases, animal from the human body, we cannot produce antibody or antitoxins inside our blood. If we happen to get snake bite, or if we happen to contact with any toxins, our body cannot fight with it. In such cases, we develop certain the blood plasma of certain very healthy animals like the horse is used to develop serum. We may inject the snake venom to that animal body, and that animal body, that such strong animals can produce antibodies in their blood. So separating that antibody, and that is used as the medicine for snake bite. Such a serum or antitoxins or antibody produced in the body of other animals, that is taken as serum, and they are used in human body. In human body it is used. Then toxin meaning poisonous protein released by pathogenic bacteria. Certain that can cause allergy, or that may lead to death in many human being. In such cases, we are developing from using other organism, we make the antitoxins or antibody, etc. Then here we are developing these antitoxins or antibodies are produced by this microorganism. Here about the snake bite treatment involve injecting the particular antivenin produced from the blood of horses, etc., or in the same manner as given above. Then again using microorganism, here another example is given. By genetic engineering has made it possible to introduce a human gene in certain bacteria, Escherichia coli, E. coli bacteria. This bacterial culture grow very rapidly. The particular gene-dependent products are extracted from this bacteria. This hormone insulin was the first such a substance produced. Usually the insulin injecting or used in the medical field is produced from the bacterial body. By genetic engineering, human gene to produce insulin is introduced into the bacterial body. Thus, with the help of bacteria, insulin is produced in large scale. Then, so such microorganisms are used in the field of medicine. Then again blood clotting factor, another serum compound produced. Here blood clotting factor 8 for treatment of hemophilia. Hemophilia is a kind of disease in that blood will not clot in certain persons. Hemophilia for clotting, we know that in cases of injury, we know that after flowing of the blood, after some time the blood will clot in the human body. That is in normal human body. If the injury is very small, the blood flow will stop itself by clotting. But in case of hemophilic persons, blood cannot clot. That is the disease hemophilia. In such cases, for clotting, a particular gene should be present in the human body. In certain persons, this kind of gene will not be present in their body. So, for treating from the microorganism, this kind of clotting factor will be produced. That may be injected to produce that blood clotting factor. So in bacterial body, in microorganism, it is produced. Then factor 9 for the treatment of hemophilia. Such many things are produced in the medical field. Then vaccine. Vaccine meaning a preparation consisting of weak germ or dead germ substances. That is also injected in, you know that weak germs, if our body, we know that for polio, tetanus, for many diseases, we are taking vaccine, especially small children. We are giving vaccines for many diseases. For the whooping cough or for the tuberculosis, BCG, etc. are the vaccines given to the newborn babies. In such cases, vaccines are prepared by using the weak germ or dead germ. We know that if it is weak germ is injected into our body, our body can fight with that bacterium and can destroy it. Then a antitoxin or antibody may be formed inside our body, so that if we get a strong germ infection, then our body will be able to fight with that strong or fully virulent germ. Our body can fight with it. So in vaccines, usually dead germs or weakened germs are used. So in vaccination also, for producing vaccine also, many this kind of bacteriums are used. Then two common vaccines obtained by growing bacteria are killed bacteria. That is TAB vaccine for typhoid and living or weakened germ for BCG. BCG is the vaccine for tuberculosis. ക്ഷയരോഗത്തിനുള്ളതാണ് BCG vaccine. Then toxoids are inactivated toxins of particular bacteria. So in the medical field, different kinds of bacteria are used. Then again, you have to know about the bacteria, its role in agriculture. You know that in agriculture, we know that many dead animal and plant body is decayed by the bacteria. So bacteria can increase the soil fertility. We know that a special category of bacteria known as rhizobium that usually live in the root nodules of the leguminous plant. Leguminous plant എന്ന് പറഞ്ഞാൽ പയറ് വർഗത്തിൽപ്പെട്ട ചെടികളുടെ ആ റൂട്ട് നോഡ്യൂൾസ് ഇവിടെ കാണാം. ഹിയർ സ്മോൾ bead like structure. In that, small bacteria are present. Rhizobium bacteria is there that can absorb the atmospheric nitrogen. We know that for production of protein, plants need nitrogen. Nitrogen is a component of protein. You know that usually the leguminous plant, പയറ് വർഗത്തിലുള്ള ചെടികൾക്കാണ് കൂടുതൽ പ്രോട്ടീൻ ഉണ്ടാക്കുന്നത്. So they have this rhizobium bacterium in their root itself, so that it can absorb the atmospheric nitrogen, so it can produce enough protein in their body. That is nitrogen fixing bacteria. That bacteria is known as nitrogen fixing bacteria. Then another bacteria is nitrogen free, that can use the free nitrogen of the soil and convert it into ammonia with hydrogen, which in turn gets converted into amino acid and nitrates, enriching the soil. Then nitrifying bacteria is there. Then nitrifying bacteria is there. This nitrifying bacteria, plants need nitrogen for synthesizing protein. They cannot make use of free atmospheric nitrogen. They can obtain it only in the form of nitrate, which they absorb from the soil through the roots. This bacteria, nitrification meaning from the dead and decaying material, this bacteria, you know that the waste of animals and dead remains of plants and animals are converted into ammonia. Ammonia is then converted into ammonium compounds. Then this is helpful for the plants. You can, which in turn first converted into nitrates by bacteria Nitrosomonas, and then into nitrates by bacteria Nitrobacter. So different kinds of bacteria here. The nitrogen cycle is given by a diagram. You can see atmospheric nitrogen is there. Then biological fixation. Rhizobium bacteria, you know that it can absorb nitrogen. Rhizobium bacteria. So it is converting it into nitrates and nitrites that can be used by the plant. So it can be used by the plants. Then plants, from the plants it will convert or go to the animal body. When animals eat the plants, this will get into the animal body. When plants and animals die, after death, the decay of the living matter, that will again change into nitrates and nitrites. So then after the death, again the denitrifying bacteria, this denitrifying bacteria is also present in the soil, that will change or make free this nitrogen and send again it into the atmospheric nitrogen.